So, welcome to the next lecture, Introduction to System Biology, and it will be about signal transduction and how we can model the principles of signal transduction. So let me first explain the essence of signal transduction. So let's say we have a cell, and we know this cell has a genome. And on this genome there are genes located. And these genes, of course, they code for proteins, and some of these proteins may be stress protected, involved in stress protection or in metabolism. Or in the secretion of certain toxin molecules by which this bacterium would fight against other bacteria. And so gene expression, as you know, is regulated by transcription factors. So there are transcription factors in the cell that regulate these genes. So for instance, they activate this one and inhibit these other two. And very often this type of gene regulation results as a response to the environment. So that means a cell needs to have machinery by which it senses its environment and by which it induces a response. So signal transduction is composed out of several steps. So one is environmental sensing, and then the second is the induction of a response, and this response is often gene expression, leading to the adjustment of protein expression and leading to different behaviors of this cell. So it can become motile, it can become stress tolerant, it may change its metabolism so that it grows on different nutrients, or it may go in some sort of fighting mode where it excretes toxins and fights other cells, or it may differentiate, so it may differentiate into a dormant cell or a spore, and etc. But all these decisions are being influenced by the environment that needs to be perceived by the cell via environmental sensing, and then a decision needs to be made leading to a response, and this entire process is called signal transduction. Okay, so that's what we're going to discuss in this chapter. Okay, so signal transduction typically starts at the membrane. It doesn't mean that all signaling processes are reliant on proteins in the membrane, but many of them are. So let's start with that. So if this is a membrane, and here there is this protein in it, a signal transduction protein, and there is this signal, clearly then the simplest process that you can distinguish is that the sensor in the membrane binds the signal. So that's the first thing. So now the outside of the receptor is altered, but ultimately, of course, we need to have changes inside too. So what typically happens is that as soon as this receptor is bound to a signal, intracellularly it changes its conformation, such that an intracellular protein can bind to it and activate other intracellular signaling processes, ultimately leading, for instance, to changes in gene expression. Okay? So that is the essence of the process. So we have, the first one is signal binding, And then here we have a second basic process is a conformational change. And here the conformational change was induced by binding, right? And but there is also another way by which cells can become, by which proteins can have conformational changes, and that's by another well-known phenomenon. And that is, for instance, so let's say we have a protein here that is this circle, and there could be a kinase that phosphorylates it, and a phosphatase that dephosphorylates it. So we have a kinase. What does this kinase do? It removes the phosphate from ATP, this gives ADP, and then the phosphatase can remove the phosphate and return the protein to its unphosphorylated state. This is typically an active protein, and this is typically an inactive protein. And the essential feature here is that the protein changes conformation upon phosphorylation and upon dephosphorylation. Okay. So this is another way by which proteins can become activated. So here this will be the active protein, and here also we will have an active protein in the membrane. Okay, so we need to understand these two basic processes. So conformational changes induced by binding and conformational changes induced by phosphorylation. So this is the first half of the chapter on signal transduction, and this is the second half of the chapter on signal transduction. And in order to do that, in order to carry out analyses, we need again the same type of machinery and terminology that has been introduced in these previous chapters. Okay, so what we need to do is we need to write down a differential equation. We need to make a model, right? That model needs rate equations. So there are certain processes, so binding processes and phosphorylation and dephosphorylation, reactions that occur at certain rates, and so we need to find these rate equations. And then we need to figure out whether there is an equilibrium state or a steady state. And we want to see how this equilibrium state or steady state depends on the signal concentration. Okay, because one way to think about signal transduction is that there is an input, that is the signal. So it's signal concentration, and there is a certain response. So there's a certain response, okay? So this is the input, and this is the output. So you can think of signaling in terms of an output versus input diagram, okay? Right, so for instance, the input is the signal concentration. So that is the molecule that, say, activates the kinase, or that's the molecule that binds to the membrane receptor. And there's this output, and that is, say, the concentration of the phosphorylated protein, or the concentration of the receptor that has changed in conformation. Now clearly there is a maximum concentration, because the maximum concentration is the maximum concentration that you have of the square protein, and equally it is the maximum concentration that you have of the membrane receptor, right? So you don't have an infinite amount of these proteins in the cell, these values are finite. So the maximal response is given by these maximal concentrations. And now there will be a certain relationship of one of these concentrations with the signal. So it could look like this, but it could equally well look like this, or it could be more linear, or it could be more step-like, or it could be more hyperbolic. Okay, so you have all these possibilities, and clearly these different input-output diagrams, they vary in terms of the sensitivity with respect to the signal. So if we would in all cases look at, say, half maximal activation, right? So we have 50% of the max. Then what you immediately see is that this curve, that the curves get steeper and steeper and steeper when you go from left to right. So this is a mechanism that is highly sensitive to the signal, whereas this mechanism is a lot less sensitive to the signal. Now what will turn out later in the chapter is that the diagram in which the protein was phosphorylated, so by this kinase and phosphatase, can lead to all these different types of sensitivities with respect to the signal, whereas the system that only has a conformational change due to binding is always hyperbolic. So it's less steep than this particular case. So this is hyperbolic curve, or this is more steep curve. Okay, so this is steep and highly sensitive, right? Whereas here the response is more gradual. Okay, and it becomes more and more sensitive, the response with respect to the signal, when you move from this mechanism to that mechanism. Now, this particular biochemical circuit can give rise to all these different outputs, and that's what we're going to see at the end of this chapter. And first we're going to start with the binding case. Okay? But in each of these cases we have to ask these questions. We have to solve these puzzles. So what is the differential equation? What is the model that we're going to analyze that captures the biochemistry associated with the signaling phenomenon? What are the associated rate equations? Then we have the differential equations with the rate equations, and we can ask ourselves, okay, is there an equilibrium state or a steady state? And in some cases we will always show that in fact these states are stable. And then when we know this, and you can ask, okay, so what about the sensitivity of this steady state with respect to the signal concentration? And then when we have that, we can draw these type of curves. Okay? So essentially what we're going to achieve in this chapter is when we go from the biochemistry, from the circuit, we go to essentially the function of the circuit. Okay? And in order to get there, we need to take these 4 independent steps. And essentially all the tools that we're going to apply in the remaining chapters have been explained in the previous chapters. So now it's essentially the only task that we have is to use the knowledge that we've learned, that we've gathered in the previous chapters. So if you understood the previous chapters, then the next ones are going to be fairly simple. Okay, so let's start at the beginning. So we have this receptor, and a signal is going to bind to it, and then we have the receptor in the membrane bound to the signal. Now the first thing that we're going to assume again is that the signal concentration is fixed. Okay, so we're going to make this assumption that we've made before, is that the signal concentration is effectively in excess. So that means that since we have fewer receptors than signal, whenever there is a signal binding to the receptor, this doesn't influence the free concentration of the signal. So the signal concentration is hardly altered when a few of these signal molecules bind to the receptor. That is because they're in great excess, and then we might as well consider them constant. Okay, so this is the first reaction that we have to think about, and this is the binding process. Right? Now, the second step is that this complex changes conformation. Okay, so that's what that is what we start with. We have this information, and then what we would like to do is that we would like to draw the concentration of the active conformation bound to the signal as function of the signal concentration. Okay, that is this input-output relationship that we are after. And yeah, so I also do this in the syllabus, so I'm going to take a slightly different approach here. I mean, the same end result, but a slightly different way of explaining it, so maybe that helps. Alright, so we have two reactions. They're both reversible, and we have a whole bunch of variables. Okay, so what are our variables? Okay, well, these are the variable concentrations R, Rs, and R star S. Okay. And again, we're going to denote the concentrations of molecules differently than their name. So the name is in capital and their concentrations are in small letters. That is because the name is associated with different properties. So this molecule also has a size and a certain weight, etc. But here we're looking at one of its characteristics, which is its concentration, so that we're going to denote by a small letter. But there are also parameters. Okay. And so for instance, the concentration of the signal is a parameter, right? So that's something that we consider fixed. And there's also the forward rate of the first reaction, the backward rate, the forward rate of the second, and the backward rate of the second. And then there are also a number of parameters that we don't mention. So they don't matter for the moment in our description, but if we would change these parameters, it would alter the description. So like temperature. So there is a temperature dependence of the kinetic constants, but we don't make this explicit because we're going to assume that this experiment is done at constant temperature. And there's also, let's say, a certain expression state of the cell. Okay? So maybe one of these processes is actually affected by other proteins, but those proteins we're not going to talk about, we're going to consider them fixed. So we're actually talking about some reference state that the cell is in. And that reference state includes certain membrane properties. And that's important because the receptor that we are considering is sitting in the membrane. So there are all kinds of implicit assumptions being made that we're not going to be very precise about at the moment. But these assumptions, they ultimately influence the values of these kinetic parameters. Okay, and now there is also another thing that you probably immediately see, and that is that there is no change in the total amount of receptors. So there is no synthesis of new receptors, or there is no degradation of receptors either. So the receptor is only going to change between three different states. So it's going to be inactive, so free, bound, and active. Okay, so there is this other constraint that we can exploit, and that is the total concentration of receptor, total receptor, that equals the free concentration plus the signal bound concentration plus the conformationally changed concentration. Okay, that's something that we can exploit. And alright, and now there is this other thing. Now we can think of the model, right? So So let's see how many variables do we have. Well, we have 3 of them. So we have dr dt, we have drs dt, and we have dr star s dt. So each of them describes the change in the concentration of either the R species, the RS species, or the conformationally changed species. And these concentrations change because of the activity of these reactions. So R is being synthesized, or consumed in the first reaction. RS is made by the first, consumed in the second. And the conformationally changed state is made in the second. Okay, and now you immediately see, you can now deduce that this relationship is actually true, because if we sum the concentrations, all the rates of changes in the concentrations, if we sum those, and since we know that the total is constant, right? It cannot be that if each of them change, that there is a net change. That's impossible, because this sum equals the sum of the total. Right? So if we add these all up, then you get minus V1 plus V1 minus V2 plus V2, and that is indeed 0. So this tells you that the total amount that we have indeed doesn't change, right? So rather than by postulating this, so like what we did before, we looked at the logic at the circuit and then we concluded this, so we postulated it, we can also prove that it's true, and this is in fact the proof. So we've just proved that indeed the total amount accepted remains constant. Now there is this other thing to think about. So if we would put this reaction in a test tube, then you can think about what happens over time to these concentrations, so R, Rs and R star S. And then I guess that by now we will no longer be surprised is that, okay, the S concentration will go down, the R concentration will go down, and Rs is formed, and then Rs star is formed. Okay, it will look a little bit like this perhaps, and the final state is going to be a state in which all these concentrations are constant. Okay, so we know that, we could check this, I do this in the syllabus, but I'm not going to explain this here. And so this final state, we have to ask ourselves, what state will it be, okay? So is it going to be an equilibrium state or is it going to be a steady state? Now, this you can deduce from the differential equations of the concentration. So if this concentration is fixed, right, so it's not fixed but constant in time, in the end state, then V1 must be 0. If V1 is 0, then V2 must be 0 too. Okay, so from this analysis of these differential equations, and by looking at the requirements for them to be 0, we conclude the only possibility for a constant state, right, is an equilibrium state, because the concentrations can only be constant if this is 0. If they would have been unequal to 0, they would be in steady state. Now, if the end state is described by thermodynamic equilibrium, like we've just proven, then we can calculate what these equilibrium concentrations, of course, are. Okay, so we have 3 unknown equilibrium concentrations. So we have 3 unknown concentrations R, Rs and R star S, and we're going to give them a subscript to denote that we are in equilibrium. And so we need three equations to solve these three unknowns, so we need three equations. Well, one is V1 is 0, that is going to be one of our equations. The second one, V2 is 0, and the third one is going to be the conservation equation of the total amount of receptor. Okay, so if V1 is 0, then K1 plus times RE times S equals K1 minus times RSE. And if V2 is 0, then K2 plus times RSE equals K2 minus times RSE star. Okay. And so using this, what we can try to do is now to solve all these equilibrium concentrations. So let's first start with solving for RE. Okay, so this equation tells us that RSE equals RE times S times K1 plus divided by K1 minus. And we know from previous chapters, that you can actually define a dissociation constant. So let's call that dissociation constant Ks. So that is the affinity of the receptor for the signal S. Okay, so this means that we can already equate RSE in terms of S. But the next thing is to equate this guy in terms of S. Well, the first thing we can do is we can equate this concentration in terms of that concentration. Okay, so and how does that go? So that is Q times plus minus. And again, we can associate an equilibrium constant to it, and this equilibrium constant we're going to call L. So that is typically done in the field. So this is an equilibrium constant for a conformational change and these are typically denoted by the letter L instead of the letter K, so that's why we're going to call them L. And now here we have RSE, we already had RSE, so this means we get this. All right. So that's what it looks like. So this allows us to solve RE. So what is that? Well, that is R total divided by 1 plus S divided by Ks and 1 plus 1 over L. Yeah, that's correct. Okay, so that is the concentration RE. Now what is now RSE? Well, that is RE times S divided by Ks, so this is going to be equal to RT S divided by Ks, 1 plus S divided by Ks, plus 1 plus 1 over L. And what is R star SE? Okay, so that is S Ks times RT, 1 plus S Ks, 1 plus